information. And that will include looking at some assessment examples. So we're going to think about some products that you're familiar with, some practices, and even some phenomena that you associate with your work as arborists. And that'll take about half the time. It's just kind of giving you an overview of that. I could literally go all day talking about this because I've been doing this myth busting and publishing on actual science as opposed to myths for a couple of decades now, but you're only getting it for half an hour. And then we're going to discuss what happens when peer review fails. And that's going to take about another half the time, and with some specific examples of things that you may have heard, actually articles you may have read, and how you can do, your due diligence in figuring out if it's good science or not. And then again, we'll have time at the end for questions that are either on this talk, or if there's things that you're curious about that I didn't get a chance to cover, that's fine as well. So I want to first of all point out that I have a extension manual out there that a colleague and I published together, which is called Scientific Literacy for the Citizen Scientist. So it's how to do this kind of myth busting, or at least assessing the validity of information that you find anywhere. It might be in the media, it might be in writing, it might be online. How do you know that what you're hearing is good information? So at the nucleus of this is something I call the CRAP test. And so the CRAP test involves taking your raw material, so your information, and putting it through a sieve, which in sieve I call the credibility/objectivity sieve. And then you might get gold, or you might get crap. And what CRAP stands for is credibility. And that means the credibility of your source, either the publisher or the author. The relevance of the information, and that means, is it relevant to you as an arborist, or is it something that has to do with crop production or something else altogether? Is it accurate? In other words, is this really old information, or if it's new information, is it substantiated by known science? And finally, what's the purpose of somebody telling you this information? Is it educational, or are they trying to sell a product? So as you can see from the beginning of those words, the acronym is CRAP. And it's an easy thing to remember, and it's obviously relatively accurate. So first of all, we'll talk about how you go through the assessment, and I will show some examples as we do this, but again, I can't do all of the things that I'd like to do. So first of all, we have things that fall under the title of no supporting science, and unfortunately, most things that I see either as practices or products kind of fall under this category. And it means either that there's no research at all, and it's just a bunch of marketing, or it might be that there's research, but it's inconsistent, meaning that some people get positive results, some people get negative results. Some people, when they do the replication, get both. And when something's inconsistent or negative in terms of the science, then at least I, as an educator, can't recommend it because it's not good, robust research in support of whatever that practice or product is. And then sometimes there's poor quality research or reporting. And what I mean by that is that you've got people doing research that have no business, pretending to be experts in the field that they're publishing in, or they publish it in a journal that is a predatory journal, or what we call a pay-for-play journal. You can publish anything you want as long as you pay for it. And so you have to be really careful, not just to look at the information that's there, but who it is that's publishing it, who it is that's writing it, and be able to kind of suss out whether it's good, bad, or otherwise. Second category is what I call misapplied science. And so these are practices and products that at least at one point were associated with agricultural production, so producing an annual crop. Many of these misapplied science products and practices aren't even used anymore. For instance, no-till agriculture is kind of supplanting the usual kind of crop tilling, just because of the impact on the soil environment, and also because it increases carbon dioxide in the atmosphere. So even though these are things that used to be things that were used in agricultural production, they're still sometimes recommended for use in arboriculture, and it's not a good fit. And then finally, we have over-extrapolated science. And so over-extrapolated science is products and practices that have limited efficacy. And that means that they just don't work very well outdoors. They might work great in a lab, they might work great in a greenhouse, but they don't work once you get outside. And this also includes phenomena, so things that we see happening in the landscape, that we can show happen in the lab or under controlled conditions, but they don't happen outside. And those are really the ones that are most fun for me because they require, a little detective work to figure out, in the literature, are these things that actually are things we worry about as practicing arborists. So we'll start with no consistent, reliable supporting science. And I will tell you that the handout that Sophia's provided you from me has the text of this talk plus extra bonus material. So you don't have to worry about taking a lot of notes or anything like that because everything that's in here to a certain extent is in that handout. When we get to the examples of where peer review kind of falls apart, that's not in the handout. That's something that I'll just talk about. So the products that we can say, pretty definitively we don't want to recommend are use of balanced fertilizers, compost tea, soil conditioners, kelp products, anything that says it's superior because it's organic and as opposed to something that's conventional, vitamin B1 transplant fertilizer, and wound dressings. And so these are things that some of them are marketed very heavily, but they aren't supported with robust science. And so I don't recommend them. And my suggestion is that all of us as, or at least most, I don't know the audience, but I'm assuming that everyone is a certified arborist. Or even if you're not certified, you want to have your clientele be able to trust what you're telling them. The practices that fall into this category are things like the recommendation not to water when it's hot outside, biodynamics, companion planting, fertilizer injection, whether into the tree itself or into the soil, hugelkultur, lasagna mulching, leaving root balls intact when planting trees and shrubs, the superiority of native plants over introduced plants, and finally permaculture. So a lot of these, as you can kind of tell from the way it's described, appeals to emotions. Things like lasagna mulching, it's lasagna, so who doesn't love lasagna? And things like hugelkultur, because they're, it's a German word, and it kind of has this gravitas behind it, that it's this old lore that's been lost, and now we're bringing it back. So, but I can tell you that for all these things, there just is no science supporting their use. And so these are things that I avoid recommending because they're not going to do any good, and sometimes they cause more damage than anything else. So the first thing I want to touch on, and I still get pushback on this, and I suppose I always will, which is fine, is that native plants are superior to introduced species. And so the example that I like to use, and I'm in the Pacific Northwest, so one of our favorite plants ornamentally is slough. And when you see it in its native habitat, which is understory of conifer forests, it looks like this. It's this beautiful, glossy green evergreen shrub with beautiful little pink flowers, and then you get these purple berries that are edible, plus they're good wildlife, and it's a lovely plant, and it gets used a lot. The problem is when it's used in an area that it's not adapted to, it looks more like the picture on the right. So it gets a lot of scorch, it gets a lot of drought stress, it gets a lot of opportunistic diseases. It's not an attractive plant at all. And this is because it's not any longer in an understory of a coniferous forest. It's now, in a traffic strip or something like that. So it's just not in its habitat. So what we have to remember is that urban sites are not natural. There are no plants native to urban sites, period. They're completely different than whatever environment these plants came from. And it's because we have issues like air pollution. We have increased drought because these urban areas are usually hotter and drier than areas that are native, that have a canopy over them. So we've got issues with heat, we have issues with increased light, we have issues with salt, and salt can come from fertilizers, it can come from construction, it comes from de-icers. The soils are nothing like what was there originally. And that's kind of the crux of the whole thing, because if you don't have a plant that's adapted to poor soils and it prefers something that is, much different, it's not going to do well because it's not going to have an established root system. And then finally, because of all these stresses, you have an increased number of pests and diseases, the opportunistic things that come in and cause problems. And that's when your clients start seeing problems. So they see browning, they see spots, they see bugs, and they're convinced that pests and disease is causing the problem, and so they're going to be wanting to use pesticides, which is just one more stress on these plants. So it's not an issue of pests; it's an issue of making plants so susceptible to pests and disease that they finally succumb to those pathogens. So what I did is I went through the literature, about 145 articles that I went through that looked at how biodiverse landscapes were if you used native plants only, or if you used, a mixture of plants. And it came from a lot of different journals, people looking at different things. So there's ecologists and there's urban foresters and a lot of different people looking at this question. And so what I did was collect this information, analyze it, and come up with, kind of a action plan, if you will, for arborists and people concerned with landscapes. So just briefly, what I found with this is that if you select properly, suitable non-native trees and shrubs, and by that I mean they're not invasive and that they are things that will live in your climate, that these properly selected non-natives will tolerate urban conditions, they support wildlife, and they enhance biodiversity of urban landscapes. And they're beautiful. I mean, there's things that we use in the landscapes wherever we happen to be that we know are non-native, they're not invasive, and they add a lot to the landscape aesthetically, as well as providing habitat for desirable wildlife. So that's kind of the bottom line, that you can use non-native trees and shrubs as long as they've been properly selected, and that's both to be non-invasive and to tolerate whatever conditions that you have. And so I think the pushback that usually happens is that people think this is what I say, and it's not. But I do recommend it, but it's after looking at the literature of a lot of different scientists from all over the world to see what they found when they were looking at this question. So as part of this, and this is something you can see with your own eyes, the push against or the push for using natives only negates the fact that native pollinators and other beneficial wildlife aren't these static populations. They evolve. One of the basic tenets of ecology is that when a new resource comes into an ecosystem, there are going to be parts of that existing ecosystem, the communities of animals that live there, that learn how to use that resource because nobody else is using it as a source of food or whatever. So this example here is of a bumblebee in Washington State that was thought to be extinct in our state, and it's the white-bottomed bumblebee. And you can see from its bottom there that's got white stripes rather than yellow stripes. And this picture was taken about 15 years ago, I think, or maybe even 20, by an amateur photographer who saw this bee and thought it was pretty cool looking, took a picture, sent the picture into the Department of Wildlife in Washington State, and discovered that it was this white-bottomed bumblebee. And you can see what it's on. It's on Himalayan blackberry, which is a listed noxious weed in our state and just tends to take over landscapes. So this isn't to promote the use of blackberry, because I don't promote its use at all, but it's to point out that this population of bees evolved to learn to use this resource that wasn't there before, because it's a great source of pollen and nectar. So that's what they do, and that's what populations of organisms do when new resources come in. They learn to use that, and we know that blackberry gets used a lot by birds and by insects because they've learned how to use this very valuable resource to them. May not be valuable to us, but it's valuable to them. So what the literature said in this particular study that I did, in general, that the most important things for a landscape to encourage the greatest biodiversity of birds, of reptiles, of insects, of mammals, was to have these three things. Diverse vegetation, that means lots of different species, not limiting the plant palette to just natives, because again, there's nothing native to an urban site. So you can choose natives that will tolerate urban conditions, but then you should bring in other things that aren't native, but that will enhance the biodiversity of the landscape. The second thing is vertical structure. So you need to have big trees and smaller trees and big shrubs and smaller shrubs, ground covers, herbaceous perennials, all kinds of stuff because there's different animals that use those resources. And so that's important, is to have that structure, especially for nesting habitat. And finally, excuse me, having the food and nesting sites. I include snags or dying trees and not taking them out completely, but leaving them for the very unique animals and microbes actually that will colonize those dead and dying structures. So those are the things that are most important, not nativity. There are, of course, certain types of wildlife that are very specialized. And this, an obvious one would be something like monarch butterflies that only use milkweed, but those are the minority of species that we see in urban environments. The majority of species can use many resources. So if you're curious about this, I first published the literature review itself in Arboriculture and Urban Forestry in 2015. And so that goes through all the articles and, in a lot more detail than I'm going through. And I also published a fact sheet. And this is the type of thing that you can share with clientele or use yourself. And so all of my fact sheets go through peer review at Washington State University. So it's a peer-reviewed publication. It's a free download. And it has, it makes the science very accessible, and it comes with a list of, action items that people can do in terms of enhancing their landscape biodiversity. Okay, so then we'll move on to misapplied science. So the products that fall into this category are things like anti-transference, Epsom salts, gypsum, hydrogels, and phosphate fertilizer. So again, there are reasons to use these things, especially in production agriculture. There are very few reasons, if any, to use them in landscape maintenance or tree care. The practices that fall under here are things like amending the soil before planting, and by amending, I mean working things into the soil. And the second one is the use of foliar fertilizers as a way to fertilize plants. So what I'm choosing to talk about is the soil amendment question, because it still tends to be an issue because there are things that are perpetually promoted as being great soil amendments, especially dealing with drought or urban stress or whatever. So we need to look at this carefully, because when you amend the soil, like working in organic matter to improve it, which again is something that we do agriculturally because we're removing organic matter from the soil when we harvest, you can tell from the picture that what's going to happen is that those two camellias are going to die because their roots are being ground up along with the soil. So we don't do this, because when you do amendment like this, you can only do it one time in a landscape, because if you try to do it again, you're going to kill everything that's there. So what we have to do is kind of mimic what happens in nature and how nature gets organic matter into the soil. And I'll discuss that at the end of talking about this problem. So besides the obvious killing of existing plants, there's some long-term effects as well. So the first is hydrology disruption. And that just means water's not going to move the way it used to when you do this kind of amendment. And so when you see standing water, like you do here, that means that there's something that's preventing it from moving through the soil. And we'll talk about what that is. Then there's soil subsidence, which happens, over time as the organic matter disappears from that amended spot. And then we have nutrient toxicities that can occur when you have too much organic matter, and it can cause overloads of particular nutrients. So briefly, what happens in terms of hydrology is depicted here in this old experiment that was done back in the 1950s by who would have been a colleague of mine, except I was not even born yet, at Washington State University. And this soil scientist looked at what happened when you would have two different soil textures touching each other. So the top, we have silt loam, so really finely textured soil sitting on top of sand. And then we've got an Erlenmeyer flask here that's dripping water into the system. And as you can see, the water gets into the silt loam, it moves every direction. So it's moving horizontally as well as vertically. And it keeps on doing that until it reaches the interface, and then it stops. And this is what always amazes your clients when you tell them this, because they think because the sand is coarser that the water is just going to pour right through it, and it doesn't. And it's because of the way that water acts with itself and with the way it acts with soil. So it binds to itself, it binds to soil, it moves through. And when you get to a different texture, all of a sudden it stops moving through because the texture is different. It'll still move sideways because the texture is the same, but it stops until you get so much weight there from just the mass of water sitting on top of that interface that it finally pushes through. But as you can tell from that third picture, what you've created is a perched water table. And that's what you see when you see water sitting on top of a soil where perhaps a lawn has just been laid down, because there's a lot of amendment and a lot of compaction that goes on when sod is in place. And that's not good for the roots of existing plants, obviously, because now you have a soil that's full of water and no oxygen. So the second problem is subsidence. And when you amend a soil and work in a lot of organic matter, you are artificially enhancing how much organic matter is naturally in the soil. And it's going to decompose as organic matter does. When it decomposes, it's not replaced because you can't go in with a rototiller and work more in. So what happens is the soil sinks, and you can tell that dark line is where the soil used to be, and it's gone down several inches from where it was when it was put in. And you can also tell by looking at the root ball of that Japanese maple, which is a clay root ball, it doesn't subside, and it's now sitting on top of the soil surface. So this will happen every time. And you see the same thing again with things like turf, and you'll see the lawn is kind of sloping away from driveways, for instance. And that's because of the settling after the soil, the organic matter decomposes. So that's the second problem. The third one has to do with organic matter and the fact that it is full of nutrients, and that you can over-amend your soil and then increase the toxic levels of certain nutrients. So when you have a soil test done, and by the way, soil tests are incredibly important to do at least once with any landscape that you're managing, whether it's yours or a client's, to find out where you are. It's kind of like going in and having lab work done at a doctor. You need to know your baseline. So if you have nutrients that are above optimum, and you can look through the chart here on the left-hand side and see that all these things are above the optimum range. And we look at the things like phosphorus. One of the keys to seeing phosphorus toxicity is when you have interveinal chlorosis. And so that picture of the rhododendron that I showed you earlier with the green veins and the yellow tissue in between, it's actually an iron deficiency in the leaves. But when you look at a soil test that says your phosphorus is excessive, but your iron is in a good spot in terms of being in the optimum range, then you know that your soil has plenty of iron. But what's happening in the soil is the phosphorus is interfering with the ability of the plant to take up the iron, and that's why it's deficient. And this has been demonstrated over and over and over through published research. So we don't want this situation. So if you have a situation where you have toxic levels of nutrients, do not add any compost, because it's just another source of those nutrients. And this is something that you can read more about in an article that I published with Jim Downer, and some of you that are located in California are going to know who Dr. Downer is. And so we talk about this, about the problems that occur when you amend the soil in terms of its structure and function. And so that's a more, and again, peer-reviewed article that goes into more detail along with a lot of references. Okay, so then we're going to move on to over-extrapolated science, and my favorite one. And so the products that fall under here, there's not a whole lot yet, but there's things like corn gluten meal as an herbicide. There's things like harpin, which is hard to find anymore, but it was touted as a way to turn on a plant's natural defenses. And then probiotics. And so this includes things, of course, like mycorrhizae, as well as beneficial bacteria. And what I mean by over-extrapolated is that you can get results when you use these products and you're testing things under controlled conditions in a lab or a greenhouse. But when you try to use them outdoors in real life, they don't work. And the second thing, the phenomena, and this is where I've kind of just dipped my toes into this, but it's turned out to be pretty fascinating, are things like allelopathy, especially black walnut trees, and humus formation. And while I'm going to talk about the allelopathy in detail, I just want to let you know that humus is something that doesn't occur in nature. And so this is one of those things that people think they're building a humus layer of recalcitrant organic material, and it doesn't exist in nature. So these things are just really fascinating to me as a scientist, because we can see how people have kind of jumped from something that occurs in a lab situation and then assuming that it's going to happen in a natural situation as well. So because lots of us are dealing with things like black walnuts or other trees that clients or we even think are allelopathic, this is what I want to spend some time talking about. So the mantra goes that, the juglone, which is the allelopathic chemical in black walnut, is going to kill plants located beneath them. And so when clients see these big bare patches where there's no ground cover, nothing else can grow into those trees, they look up, and the first thing they'll find if they look up black walnuts is that black walnuts are allelopathic, and it's because of this chemical. And so the first thing I want to say is that juglone absolutely does exist, and it absolutely is toxic to plants. The problem is it's only in certain situations. So what allelopathy is, if you're not familiar with it, it's basically just the ability of plant extracts to kill other plants. So allelo just means other, and path means death. So this is death to others. The way that allelopathy is tested is that you take these extracts, and so you macerate tissue and you extract a particular compound, and then you add it to petri dishes on which you have germinating lettuce seeds, and you add increased concentrations of those things until they die. So in this picture here, where it's clear and these are growing on agar, you can see that everything looks healthy. It's the agar is clear and the little germinating seedlings are fine. But then you can see when you start adding a compound that you get this darkening of the tissue or of the agar, and that's because the plants are dying and all these things are leaching out of them. The problem is that when scientists discovered this ability of juglone to do this in the laboratory, and this is probably back in the 1930s, they immediately assumed that what they were seeing in a petri dish then would also happen in the landscape with real trees and real plants underneath. And that's what you call taking two things, which is correlation, and then pushing it into causation. In other words, one of these things is causing the other things. So this is taking the negative effects in these lab experiments and then extrapolating them to landscape conditions, where if you had a black walnut tree and it was near anything else, that the juglone from that tree was going to be killing those plants. And I will tell you that the reason that this even started as a thing was because one of the scientists who discovered juglone, again, and this is back in the 1930s, and this is when technology finally allowed us to separate out chemicals and analyze them and identify them, by their chemical structure and then name them. He had a black walnut tree at home, and he also had tomato plants growing at home. And he noticed that the tomato plants that were closest to the black walnut tree did the worst in terms of growing. And then as you got farther away from the tree, they did better. And so he immediately extrapolated what he'd seen in his work in lab, that, aha, that's why my tomato plants are dying, because they're too close to the black walnut tree. Well, this whole allelopathic thing was a huge scientific advancement. And, agricultural scientists are very excited, because if you can find these types of compounds, then you can make natural herbicides, right? So this is something that was one of the key points from discovering these compounds. So lots and lots and lots of plants have been analyzed, and their compounds have been extracted and tested, and there's thousands, if not hundreds of thousands, of different chemicals that are allelopathic when you test them in petri dishes with germinating lettuce seeds. The problem is that a black walnut tree in the landscape is not the same thing as being in a petri dish. So there wasn't any real reason to assume that this was happening, other than it was just seeing two different events, and those two different events, that's the correlation, and then pushing them to causation is the problem. And so we're left with this vision that all that juglone's coming out of the leaves, it's being leached through with rain, it's coming out of the roots, it's coming out from decomposing husks and leaves and everything else. But there isn't any evidence that this happens consistently, because we know plants can grow into black walnut trees. In fact, this is what a lot of black walnut growers were telling the scientists in the 1930s, because they're kind of worried about this, was, we grow walnuts, and there's lots of stuff that grows under the trees. It's not a problem. I've seen a number of landscapes that have black walnut trees, and lovely landscapes underneath them. So this is a problem for the scientists now, because they have to explain, okay, why does it kill some plants but apparently not kill others? So they started making these lists, and the lists were plants that are tolerant to juglone and plants that are sensitive to juglone. And I'm a plant physiologist, so I'm looking through these lists, and the first thing that occurs to me is, there's no physiological reason that some plants would be sensitive and other plants wouldn't be. In other words, the species that were there were all over the place. There were trees and shrubs and vegetables and herbaceous perennials and annuals and vegetables, all kinds of things in both lists. So there wasn't a hypothesis out there that you could formulate saying these ones are sensitive because of, and then fill in the blank, and these ones are tolerant because of. That makes it a list that is not really very reliable. And so my goal was to figure out where these lists came from, to get back to the original research that generated the data that would make these lists. So what we found was, what I found looking through this, is that there was obvious evidence of allelopathy from juglone and everything else in killing plants in petri dishes. But there is no published research to support juglone toxicity in the landscape, not one paper. So in other words, all the research that had been done either had negative results saying, the juglone didn't do anything to plants there. And in other cases, people were looking for juglone in the soil and they couldn't find it in substantial concentrations. So they're left with no evidence that this is a problem in the landscape. So meanwhile, back my venture towards trying to figure out what's out there in literature where these lists came from, is I was able to track back to one original paper that came out of Cornell in the 1950s, and it was something called walnut wilt, and it was written by L. Christ and then another author whose name I don't remember. And so all of these papers would cite this walnut wilt paper as being the source for this information. So I have a former graduate student who's now on faculty at Cornell, and I've emailed her and I said, Taryn, I can't find this paper online. I can't find it anywhere. Can you see if your extension service still has this old paper? And they actually have a library at Cornell in extension where they keep everything that's ever been published through extension. So about 2 weeks later, I hear back and I was told there is no such paper. There's no such paper. There was no one named L. Christ ever on faculty. They have no idea where this paper came from or where this thing came from. So it's all non-existent. I mean, there's nothing there. So, and I will tell you that researchers who have done peer-reviewed publications, they will cite this paper, and I've actually contacted some of them to say, hey, I can't find this. Can you send it to me? Because I'd like to see it. And none of them have it. They'll have excuses. Oh, I've moved. Oh, well, I don't know where all that stuff is. But none of them, I'm sure, have ever seen this paper because Cornell says it doesn't exist. So it's been this big, brouhaha about something that doesn't happen in the landscape. And this is why when they've tried to use allelopathic chemicals and use them as herbicides, they don't work. And the reason is because all these things are organic materials. And as soon as you put them on the soil, real soil, not a petri dish with agar, that the microbes eat them up. So they break it down really quickly, and there just isn't enough there to cause a problem. So those plant lists are non-existent. And probably the reason that some things won't grow into black walnuts is because there's not enough water. And established black walnuts, like lots of other trees, like cedars are sometimes also tagged as being allelopathic. They're just really competitive. And so if you put in new plants around an existing, competitive tree, yeah, they're going to die. They're not going to do well unless they have more water or fertilizer, whatever it is that they need. So it's not really very sexy and exciting, but probably the reason that plants don't grow under black walnuts is they don't have enough water or enough sun, depending on what the plants are. And if you want to read more about this, I've got a publication on it. But the take-home message is that when you have two things that are correlated, does not mean that one causes the other. And this is a really important lesson to learn for lots and lots of things that clients are going to be afraid of. So there's one website I have found, it's called Spurious Correlations. It's hysterically funny. It consists of tens of thousands of correlations this guy has put together. This is one of my favorites, which correlates the per capita consumption of cheese with the number of people who died by becoming tangled in their bed sheets. So, I mean, it's a perfect correlation. So, the message here would be don't eat cheese before you go to bed because you're going to die. And everybody can laugh at that and see that it's really pretty ridiculous, but they're correlated. So you have to take it backwards and say, just because you see a correlation, you can't say it's causation unless there's published evidence to show that there is causation. And published evidence for allelopathy doesn't exist in the landscape. So as I said, this is a really important message to share with your clients. So I have a peer-reviewed fact sheet again on this. It's free download that talks about this whole story in a lot more detail. It's just a fascinating thing. And I mean, I used to teach about allelopathy when I was teaching plant physiology at the college level. I would teach it completely differently now because I've done the work to find out where the science was, and it doesn't exist. So that leads me to good and not so good science. And so there's stuff that we would consider to be good quality research, but it's poor reporting. And this is often due to researcher bias. It's not that we deliberately are biased. Bias is sometimes just completely subconscious. And it's because we're human. Even scientists are human. We have a hypothesis, we do our research, and we subconsciously want to be proven right. So what happens is that we will selectively tell you about some of the results in the paper, in the abstract, and then we might not tell you about other results in the abstract. The paper itself has that information, but as many of you know, sometimes you can't see anything but an abstract because it's behind a paywall, or it might be that you don't really have time to read the whole paper and you are trusting that the abstract is going to give you a good overall summary of what happened. And that's unfortunately not always doable because peer review sometimes does not catch this bias. And so you're going to be left with a biased message and that it will freak you out. And a case in point happened several years ago when there was a paper published that claimed that newly transplanted trees should not be mulched. So this was a shock to me and other people who also published on mulches, but it was written by Ed Gilman. And everybody, you know, Ed Gilman is the god of tree publications. I mean, he's now retired, but you know, he has a huge body of work, and a lot of that work has guided what we do as arborists. So this paper from 2012 was actually a pretty simple paper. And so, of course, those of us that are involved in mulch research immediately want to read the whole thing to figure out why this claim was out there. So the methods are real simple. They used containers that were filled up with soil, and then they either left them uncovered or they covered them with mulch. And so we have some pots that were just bare soil and some pots that were soil plus mulch on top. And then what they did was they watered them completely, you know, saturated the soil, and then measured the amount of water being lost through evaporation for 3 days afterwards to find out how much water is retained, you know, because of the mulch. And the results are pretty simple too. So in the unmulched soil, so no mulch on top, the total evaporation was around 3 liters on average. On the mulched soil, the total evaporation was 2 liters. So the results are not surprising. The results are actually, you know, supportive of what the science says right now about what mulch does in terms of evaporation. So if you were to graph this out, it would look like this. So you would have a title that would say something like mulch reduces the evaporation from soil by 33%. And then you'd have this bar graph showing that that's exactly what it did. Had this information been in the abstract, there would have been no problem, but it wasn't. So what the abstract said in part was, given minor reduction in evaporation and reported disadvantages of mulch application close to the trunk, landscape managers might consider changing mulch application practices for newly planted trees. So as a peer reviewer, and I do lots of peer reviewing, and I'm also an editor. So the first thing that peer reviewers are supposed to do when they're looking at the abstract is making sure that the abstract is a complete record of what's happening in the paper, so people do get an accurate summary. Well, we don't have any trees in this research. We have pots of soil. Trees aren't part of this at all. It's a soil experiment. So having anything that even talks about trees shouldn't be in the abstract because that's not what was tested. You could discuss this in the paper itself in a discussion section about what you think you should and shouldn't do, but it's not something that tells you what was done in the paper. So no trees in this experiment, and nothing about newly planted trees or trunks or anything else. So this was in the abstract, but the actual fact that 33% reduction in water loss was not mentioned. It was called minor, and I'm sorry, but 33% is not minor. This is the message that people that only read the abstract got. So one of my peers who was not amused with this published online this critique, which said the Florida study was conducted in a highly artificial system, was very narrowly focused, and reflected water loss over a period of 3 days. We have conducted field trials with real trees and shrubs growing in real soil, where we have found real benefits in terms of plant survival, plant growth, soil moisture, and plant water relations over the course of years. And that's where the science stands. So what you have to think about when you see papers like this, even when it's by the preeminent researcher on tree research, is don't succumb to what I call SSS, single study syndrome. It doesn't matter who writes it. If it's at odds with the majority of other papers, then it has to withstand increased scrutiny. So you want to get into that paper, you want to read the results, because science does change. It changes all the time, especially in our field. Our field is relatively young. So it could have been something that was brand new, but it wasn't. But the problem was the abstract was misleading, especially for people who weren't able to get into the results themselves. So that's kind of the message there is regardless of who it is, if it's only one study and there's not other supporting studies and it hasn't been repeated by anybody else, it's not something to change, your whole opinion about a particular topic. Okay, so I'll give you another example too, which feeds back into the native trees issue. And the claim here is that only native trees can provide resources for native insects. And this was published in 2009, and also by someone who's preeminent in their particular field, who's Doug Tallamy. And he was looking at the impact of native versus introduced plants on survival of Lepidopteran larvae. So these would be larvae of butterflies and moths, which is his big thing. So this is, and these are entomologists who are doing this work, and they published in the journal Conservation Biology. So their methods, and these are in quotation marks because this is directly out of the paper, what they consider to be native or non-native is they labeled all genera. So this is the group of, the genus. They took all of the genera that comprise native species with geographic distribution in the mid-Atlantic region. So those are where they're working, and they called all of those natives. So it's 103 genera. So this is 103 genuses, even if the genus contained one or more species that have been imported for use from Europe or Asia. Okay, well, that's a problem, because now you're calling a genus native, but you're including in that genus species that you found in your study that are not native. So it's literally mixing apples and oranges. And you can't do that when you're trying to distinguish between native and non-native plants. You just can't do that because it completely takes away your argument. You don't have a way of comparing them anymore. What happened in this was that almost 200 introduced tree species were folded into native genera. And these were things, it could be a variety of trees. I'm not familiar with the vegetation so much in the mid-Atlantic region, but I know a lot of these woody species would be probably problematic in terms of being aggressive. The analysis that they did with the data is that they took all the things they found from Lepidoptera choices and then said this would be the same for all insects. In fact, they said, we can think of no reason why Lepidoptera would not serve as a reasonable surrogate for other folivorous, and that means eating leaves, insect herbivores. So this would include, elm leaf beetle, any of these things that happen to be, that will strip Japanese beetles, you name it, and you can think of all kinds of things that are not lepidopterans, that are very general in what they eat. And lepidopterans, again, are rather specific, sometimes specific to a particular genus in terms of what they'll eat. So not only did they kind of mush everything together in terms of native versus non-native plants, they then said that this was going to be true of all insects, not just lepidopterans. And that's, that's really pushing it. So why all this stuff was allowed to get through is not really quite clear. So the results here, and this little table kind of shows what they said, the 20 most valuable plant genera includes members of Prunus, it includes members of Rubus, it includes members of Rosa, and other things, Pyrus, other things that have members that are noxious, that are invasive, but they're valuable to wildlife, which is kind of the point I was making before, is that even native, rare insects will use things like blackberry because it is so, useful in terms of what it provides in terms of a food resource. So it's kind of a meaningless paper because you can't say that native plants were better because you've included a bunch of non-native, almost 200 non-native plants in your analysis. So as a meaningless paper, they should have had a plant scientist on this to help them divide things up better so that they could actually make a rational argument comparing native to non-native rather than just comparing artificially constructed groups. So, and other people have said this as well, is that this study really was fatally flawed from the beginning because it combined all these species at the genus level and considered them all to be native, and drew all of their conclusions from this really unacceptable combination. So this is what happens when peer review, fails. It happened with the Gilman paper. We're not really sure why, but what should have happened was that the peers should have seen that the abstract did not reflect the results accurately and should have been rewritten. In the Talmy paper, again, he is a big name in insect conservation, and peer review should have noticed the flaws in the methodology and had him reanalyze the data, truly separating out the native from the non-native trees. So this is what happens when peer review flops, and this is why sometimes it's really important to have somebody, whether it's you or somebody else, kind of go through a paper and figure out is this good science or not. So those ones are kind of tricky, excuse me, because you have to really get into the data. The not so good science is a lot easier, and so if you want to try honing your skills, I suggest trying things like this because they're pretty easy marks. So with poor quality research, this is really common with people who have no experience in the field at all. They have their PhD, and that makes them think that they are experts in everything. And I can tell you as a PhD is I am not an expert at anything, and I would never choose to step outside my comfort zone or my expertise zone and pretend to be a researcher in another field. Oftentimes these authors will conflate correlation with causation, and they're almost always published in what are considered by real scientists to be marginal journals, pay for play, usually with no peer review. And so it's junk science, for lack of a better term. This is junk science, but the problem is it's really hard for non-scientists to be able to figure out what's junk and what's not, unless they have some steps to go through. So we'll look first of all at this claim, which is neonicotinoids cause colony collapse disorder. And we know that neonics can be a problem, especially if they're applied at the wrong time, but that's more on insects in the field and not colony collapse disorder. So this is a different topic. And this came out in 2014. In the Bulletin of Insectology, we've got our 3 authors here. So, it's a perfectly legitimate title. You can understand what it says, but then you have to get into the crap analysis. And so that's what I'll do here. As I said, these ones are pretty simple to do, and they're fun. So here are the article details. We'll start with the C, credibility. Okay, well, the first author is from Harvard, which is a good thing. His other authors are beekeepers, and this is not a bad thing. All it means is that the other authors, the beekeepers, were cooperating with the study. When you're doing these field studies, you have to use someone's hives. Most researchers don't have their own hives, and so they use hives of beekeepers, and they give the beekeepers author credit because without their help, they could not have done the work. So that's kind of the bottom line, is that they're authors because their presence in the experiment was critical to the experiment. Okay, the second thing to note is that the author has a PhD in public health. Okay, not an entomologist, not a pesticide expert, nothing having to do with anything on this topic, because we're not even talking about impacts of neonics on humans. We're talking about impacts of neonics on insects. And so the PhD in public health does not qualify him to be a researcher for this kind of work if he doesn't have a co-author who's an entomologist, and he doesn't. He has 2 non-scientists as co-authors, so he's the only scientist on this paper. If you read this paper, you'll notice it has poor English throughout the article. And I know it's going to sound kind of colonial, but the language of science is English, and every scientist knows that. So if you publish in a journal that does not have well-edited English throughout it, it's immediately suspect, because that's just that's just the way that science functions. So when you see this type of thing, then that's another red flag. In the research itself, the bees were force-fed high fructose corn syrup containing high levels of neonicotinoids. So for those of you that aren't familiar how entomology research is normally done, is it's usually a choice study. So it's choice feeding. Bees are given a choice of things to feed on. In this case, they had one choice, or they had starving to death. And so of course they're going to feed on high fructose corn syrup, which is not a natural food source, with high levels of neonicotinoids, which is also not something that would happen in nature. So even entomologists looking at this said that the colonies were overdosed compared to the real world, because they wouldn't be forced to be feeding on high levels of neonicotinoids in the real world. And then the capper to this whole thing, when you read the paper, is there's no statistics. Okay, so they talk about all these significant things, but they have no statistics to demonstrate that there was any effect. So this is when your BS meter goes completely off scale, because there's so many problems with it. And in fact, entomologists have roundly criticized this paper as just being bad science. So don't have to worry about that in terms of colony collapse disorder. It certainly was not a paper that would demonstrate that. The last one I want to talk about is one that really irritates me, because it makes my life miserable personally. And that's the perception that glyphosate causes human disease. So glyphosate, as you know, is the active ingredient in Roundup. And one of the first papers that came out with this claim was in 2013. The first thing I want you to notice about this is the title. And the title is designed to discourage you from questioning, because you're going to read that, and if you're not a scientist, you're to not know what half those words mean. I can tell you as a scientist, I can read it and I don't know what most of it actually means, because it's a whole bunch of words been strung together and it doesn't really tell me anything. And about why this would, it's nothing. It's gobbledygook. It's published in the journal Entropy, which is quite amusing, just to me personally. But we'll talk about why that journal is not a great journal. So this author, let me go back one step. The second author, Stephanie Seneff, is the author of all of these papers. And she works with various people and publishes this stuff and continues to publish this stuff. So because of her work, it's almost only her work, there's been all this fear mongering, they say that glyphosate will cause obesity, Alzheimer's, autism, anorexia, dementia, inflammatory bowel disease, depression, and makes me depressed just thinking about it, Parkinson's, reproductive issues, liver disease, and cancer. And you get all the memes that talk about how, your food is contaminated with glyphosate, and this is why everyone's getting sick, and it's all because of glyphosate. So you have to start taking the articles apart. So we'll start with this one because it was the first one. Well, the authors at MIT, again, not a red flag, a good thing. Unfortunately, her degree is in computer science. She has no background in any life sciences whatsoever, whether it's human medicine, whether it's biochemistry, anything. She has a degree in computer science. With that degree, she could not publish this thing anywhere in a legitimate journal. So she settles for the Journal of Entropy, which is a predatory journal. Has no peer review, and you pay and they will publish it. It has phrases like this. So you thought that if you really want to drive yourself crazy, read this article, because it is so full of, if AI would have existed back then, I would say it would be AI generated paper, because it is so full of just jargon and stuff that's designed to confuse you, that you're left saying, oh, she's a scientist and she knows more than I do, so I'll just believe whatever she says. So she says that glyphosate is a textbook example of exogenous semiotic entropy, disruption of homeostasis by environmental toxins. So that's a direct quote. Okay, I know a lot about glyphosate research. I've read a lot about it, but I've never heard of exogenous semiotic entropy before. So I took that phrase, and remember, this is back in 2013. I took that phrase, exogenous semiotic entropy, in quotation marks, and I put it into Google, because I wanted to see how many more examples there were of this thing that I couldn't even understand what it meant. And there was one hit, and the only hit was her article. So she makes this crap up. Then she says it's a textbook example. and then she publishes it. And it plays well with a certain audience, as I'm sure you know. The audience that doesn't trust big ag, it doesn't trust chemicals, it doesn't trust anything. And so one of these groups, found her online and published an interview with her. So as I said, this stuff's on the internet. I love finding it. This is what she said. So basically what I do is I read papers and I process them with the computer to help me understand them and interpret them and generalize and build a story. She's not lying. She's a great storyteller. This is not science. This isn't even junk science. It's garbage. I can't even use a science word with it. So by this point, the BS meter should be blowing up because it is the worst example of academic malfeasance that I can think of. Is someone who has absolutely no business writing any of this stuff, writes it and fearmongers, and that's what she does. So she has charts like this, where she shows that glyphosate has gone up and autism's gone up. Oh my God, the glyphosate is causing autism. Well, vegetarianism's gone up. There's all kinds of things that have gone up. Do they cause autism? She's not saying they do. She only focuses on glyphosate and anything that's correlated with the use of glyphosate. And that's what she's done in numerous papers. And it freaks people out. And that's why lawsuits are decided, is because of this kind of fear mongering, and because there are lawyers out there that are just hungry for finding someone who's got some disease and then linking it to this. And so it's not science that's deciding these lawsuits, it's really good lawyers and fear, because lawsuits are not decided on science unless the scientists have the best lawyers. So this is really frustrating to me because, for instance, I just found out that her newest thing, and you can again find this online, is she's correlating COVID-19 with glyphosate. And her reasoning is that when you make biofuels, you're using genetically modified corn. And when you use that fuel, it's putting glyphosate out in the atmosphere. And that's what made people susceptible to COVID-19. She's absolutely crazy. She truly is. And I don't mind even saying that on something that's recorded that she could hear someday in the future, because she is absolutely crazy. There's absolutely no science behind this at all. So it doesn't matter because a lot of us won't be able to use glyphosate anymore anyway for various reasons, but it's kind of too bad because it's a good tool. It's a chemical that only affects a biochemical pathway in plants. It does not affect humans or animals of any sort, because we don't have the ability to make lignin. If we could make lignin, then we'd have a problem, but we don't. So how can you find good information? Well, I have a lot of stuff online. This is my horticultural myth page, where I've got about 70 or so white papers that are free downloads, and just kind of, they're not peer reviewed, and they haven't gone through, they don't have references there, but they're all based on science. I just have them there because they're still all valid. If they were bogus, I'd take them down, but I don't do bogus stuff. So that's one side I've got. The manual is out there too, and I really do encourage you to download it and use it. It's kind of fun, it's very interactive, and it gives you some good science. I've published a number of books, as Sophia mentioned, and out of all of those, understand how plants work, as well as mythology stuff, are just really useful. So if you're, I'm not here to sell books, but just to let you know, there are good books out there that are actually based on good science. And then I've got a series that's on DVD that, if you really enjoy listening and watching me, you have 12 hours of that kind of fun. I keep all of my articles actually managed on a site that's called ResearchGate, and it's generally kind of a place for researchers to kind of share their information with each other, but anybody can create a free account. It doesn't cost anything. As I said, they don't collect your information, but it's a way to get in there. And then for anybody who's published anything, you can search for them and you can get the list of all the articles they have. And then I've loaded these up as things that you can download and use for yourself. The Journal of NACA is something that I've been publishing in along with Jim Downer for a number of years, and we've published things on managing landscape trees, as well as structure and function, as I told you before, soil nutrition, pruning woody plants, and using arborist wood chips as landscape mulch. And so every year we try to get together and publish one of these review articles for use by not just extension people, but by practitioners as well. This is our blog page, which has been up there since 2009. Every week we have a new post. I'm not the only one that posts. We have a number of people that are experts, PhD experts, that have interesting information to share. So we've got people that we've got a climatologist, Jim Downer publishes on here, people that you've heard before, just with whatever our latest thoughts are about any particular subject. We also have a Facebook page, which we kind of use as a clearinghouse to link up, good science-based information, just as a nice place to park websites and blogs. And then the last thing we have, which I really encourage you to join, is our Garden Professors blog group. And it is on Facebook, so you have to be on Facebook to do this. But the nice thing is it's got over 26,000 members, so there's somebody on there all the time. It's a science-based group that's pretty tightly managed by 12 volunteer administrators. We don't do it yourself. We don't do home remedies. We don't do anecdotes. It's just good science-based information. So you come on with a question. You can search to see if someone's asked that before, because it's been up since 2011. So there's a lot of archives to go through. And if you have a question that hasn't been asked before and you put it up there along with the information they ask, you'll get answers back immediately from all kinds of people. And we just, as I say, make sure that the information that's posted there is actually legitimate. And if it's not, we take it off. So if you decide to join us, I do want to tell you, you have to answer 3 questions. So don't just say you want to join and not answer the questions, because those questions are there to make sure you're not a bot. So you have to answer the 3 questions. They're not difficult, but that just ensures that you're a real person, and then you'll be in the group. So with that, I'd love to answer any questions that are up there. And Sophia, if you wouldn't mind reading questions to me out of chat, that would be great. And then I can just answer those without having to read and talk at the same time, which does not work well.
Yeah, absolutely. So if anyone wants to ask Linda questions directly, feel free to unmute yourself. And I know some of you have to take off soon or about now, so thank you for your time. I just wanted to say, if anyone is signing in as iPhone, just let me know who you are. You could tell me directly in the chat, just so I can get you CEUs if you need them. Because I can't do that if I don't know who you are. Okay, so let's see. So you mentioned lasagna mulching being a myth.
Right. So what lasagna mulching is, if you're not familiar with it, is using layers of cardboard and compost to make a lasagna that is designed to smother weeds. And so we've actually published some research on why this doesn't work. But briefly, the problem is that cardboard is a sheet mulch, and it restricts oxygen and water movement. And the more cardboard you have, the more restriction there is. And so there's that problem, which keeps your soil from being particularly healthy. And then again, having lots and lots of organic matter, adding onto a soil is going to potentially cause some problems in terms of nutrient overload. What we've shown with mulches, though, specifically, is that one sheet of cardboard, just one sheet of cardboard on top of the soil, is 10 times worse than 4 inches of arborist wood chip mulch in terms of oxygen and carbon dioxide transfer. One sheet. So anything that's a sheet mulch, whether it's newspaper, cardboard, landscape fabric, plastic, and those last 2 are the worst of all, of course, is going to inhibit water and, oxygen and carbon dioxide movement, and probably water as well. We didn't test water, but you can test that yourself just with a simple experiment. So that's why we don't recommend it.
Okay. Kind of along the lines of organic matter. So you mentioned humus doesn't actually exist in nature. So what exactly is going on?
Okay, so humus is defined as being this recalcitrant organic material that, organic material breaks down until it reaches this point where it becomes recalcitrant. So it just stops breaking down. It becomes more like biochar, which is organic matter that's been through pyrolysis, and it becomes more like rock than anything else. So there's been this perception that this is what happens naturally with wood. And the way that humus was originally defined was it's a product of soil testing. When you're testing for organic matter in a soil, you treat it with an alkali agent, which has a pH of 12, and it makes humic acid and other types of humus compounds, and they don't break down. But you don't have soil conditions that are pH 12. So it's something you can have happen in the lab, but it doesn't happen in nature. And it kind of makes sense. If you think about it, you have this recalcitrant material that's from woody material breaking down that's been going on for eons. Why wouldn't we be 12 miles deep in humus? You know, so it's something. There was a really good article, a review article written on this in Nature magazine, I think back in 2017. It went through all the literature, kind of like I do, with myth busting stuff, to find out if there was evidence for humus existing in landscape, and it doesn't. You know, it's everything breaks down, and we know that because we work with organic matter, especially in terms of wood chips, all the time, and we know that we have to replace it because it goes away. So it's not that it breaks, that it stops breaking down and turns into this other material. It just doesn't happen in nature.
Okay, so I've been hearing about and telling people that humus is the organic matter in the soil. It's another name for the organic matter in the soil. It's what the bacteria are building. It's where they live. So a more accurate way of saying it is just to say organic matter.
Yeah, I would just say decomposing woody material, because that's where this whole humus thing came from. So decomposing woody material does not have the word humus in it.
Okay. Yeah, lots of comments from people wondering about all the things they have to unlearn now.
Well, I will tell you that when I was doing my PhD back in the 80s, it's been a while, the information out there on practical plant physiology, on urban horticulture, arboriculture didn't really even exist much in that point. There wasn't any. And so I was laboring under many of these myths, all through my PhD, because there wasn't better information out there. So as a matter of just being able to develop the area as an area of research and generate good information that we're able to now myth bust. So that's good for our field, that we're getting good science rather than just relying on folklore.
Mm-hmm. Let's see. I don't see questions from other people. Again, if anyone has questions, feel free to ask. But I was wondering what your take on mycorrhizal inoculants is.
And that's a fun one too. So it's one of those ones that falls under over extrapolated information. So mycorrhizal and mycorrhizae are obviously important, and mycorrhizal spores are really important to have in your soil to inoculate, your tree roots. And there are trees in general that don't have mycorrhizae. I mean, nearly all wood plants have mycorrhizal species. The problem is that the inoculants themselves don't work. They work in the lab. They work great in the lab if they're alive. Sometimes the inoculants are actually dead, so there's always that problem. But let's say, for argument's sake, that if you had things that were growing in a greenhouse and you were using a soilless media and it had been completely sterilized, which is what you do when you're doing research, is you sterilize it, and you can get those inoculants to grow and to inoculate the plants. It works great under controlled conditions. Once you get outside, if you take half your plants and they're your controls and the other half you inoculate and then you plant them out, there's no difference. And when you dig them up a year or so later, everything's inoculated because there's already spores out there. And in fact, all the ones that are inoculated with whatever's native there. They're not inoculated with the species that they were originally dipped into. So it's already there. If you have a healthy landscape that has diversity of woody plants on it, you've got all the mycorrhizal spores you need right there. So you feel like inoculating something, take a handful of soil from that and sprinkle it wherever you feel like putting it. But you don't have to waste your money on packaged products.
Okay, I see a lot of products for mycorrhizal inoculants, and also like molasses and sugars to feed all the microbes.
Yeah, and yeah. I mean, to a certain extent, molasses and sugar, it's a carbon source. It doesn't help the plants any. What it does is it feeds bacteria. And I mean, if you for some reason need to increase your bacterial activity, that would do that, but it's not going to do anything in terms of helping plants directly.
Okay. Since I have you on here, I was wondering about biochar. You mentioned biochar, and I know it's becoming more and more popular, incorporating it into landscape soils. What's your take on that?
Yeah, and I actually have a fact sheet on that. I have a fact sheet also on mycorrhizal products. So if you get into my website, you can find those. But back to the question about biochar, it's one of those things. When I first heard about it, I wanted to hate it because it's a bio slash material. I don't like bio dash or eco dash or any of those things. They make me cringe. But there wasn't that much research when I first heard about it, so I decided to wait for a few years and then do the literature search and see what the science said about biochar. And it actually has a lot of benefits. It's more beneficial in agricultural systems when you're working with crops. It works better in tropical and subtropical areas than it does temperate areas. The problem with it from a practical standpoint, even though it's a wonderful material and I believe the science behind it, is that if it's used as an amendment, then you're going to have the issue with amending part of soil and not the rest of it. You change the soil texture and you get perched water tables. So I see it as being useful in things like tree wells, where you're putting in trees into an urban system or street system, where you're constructing completely new soil bed, and where you can you use a construction soil or something, and you can mix biochar into it entirely. So it's like a big container, right? So if you have it completely mixed in there, it's great. If you're trying to figure out how to do a green roof, for example, another system where you would be bringing in material that, there's nothing there, and bringing in new material entirely, you can mix into that. I think it would be great in green roofs. You can use it in containers. You can't use it as a mulch because it causes damage to the trees. And you can't just amend part of a landscape and not the rest, because you're going to have the issue with water movement. But in terms of the things that biochar can do, in terms of enhancing beneficial microbes and enhancing, water and oxygen movement in the soil, it's the list that I have on my fact sheet is long in terms of the benefits. The problem is figuring out how to use it practically.
Okay, so we use a liquid biochar and inject that into the soil. Is that better than incorporating the granular?
I have never heard of a liquid biochar, so I don't know what that would be. I'm gonna have to look that up. I'm gonna write that down.
It's not available anywhere in California. We bring it in out of a manufacturer.
Oh, that's okay. I do liquid searches internationally, so I'm just curious what it is.
It's biochar that's brown, like super fine, so it can mix in with our combo treatments, and we could do soil injections. So we usually do mix it with some micronutrients or even mycorrhizae.
So when you say it's ground fine, how fine is fine?
Do you have it's something like microns?
Okay, so then it loses most of its function, because a lot of the function of biochar is to increase soil permeability, to provide actual physical space for beneficial microbes. I mean, the way it's been.
I could send you the research papers on the benefits of they've done a bunch. I mean, again, it's the people producing it is also doing the research paper, so it'd be interesting for you to look at it and see what you can find.
Yeah, but especially, I mean, and so for a research paper, that means it has to be published in a peer-reviewed journal. It can't just be an in-house publication. It has to have gone through peer review. So otherwise, it's more or less like a testimonial. So if you've got actual research papers, that would be great. But they again would have to be things that had gone through peer review, because otherwise it doesn't meet the standard of peer-reviewed research.
Okay.
Yeah, that's new, and I had not heard of liquid biochar before. But again, most of the benefits of biochar come because of its structure and grinding it up fine.
Structure for its size, I don't know. But yeah, I'll send it to you so you could look at it. Yeah, and I'll get feedback.
Yeah, because microns are not going to do it in terms of the structure. But that's a new one.
Okay.
Hey,
There's
I need to jump off, but I just want to say thank you for this presentation and this discussion. This has been great.
I'm glad you enjoyed it. Thanks, Ralph.
Yeah, thank you. This has been tremendous.
Oh,
Yep.
thank you, Jexie. Appreciate that.
Also a PhD in horticulture from Berkeley.
I'm sorry, I missed the first part of that, Sarah. I didn't hear what you said.
Oh, Ralph, who was on there, he's PhD from
Oh,
Berkeley.
oh, neat. Very
Yeah,
cool.
so he's speaking right in his wheelhouse.
all right. Question from Jackie here. Are there any studies about glyphosate not being threatening to people? What about that lawsuit with the janitor?
Yeah, well, you can't do research on people with glyphosate because it's unethical and illegal. What you can do is do research on glyphosate on cells or on other animals like rats. And in all the research that's been done, looking at LD50s, and I think if you do any kind of pesticide application, you know what an LD50 is. So a big LD50 is a good thing, a small LD50 is a bad thing. The LD50 for glyphosate is really pretty large. And you look at other things like nicotine, for example, which has a very small LD50. It's a very toxic material. So glyphosate is just something that, because it doesn't affect one of our enzymes, it's not toxic. Now, Roundup, on the other hand, I can't tell you what's in Roundup because a lot of their inert materials are proprietary. So I don't know. I don't know what Roundup has in it, and I don't make any claims about Roundup being safe. What I say is the active ingredient, glyphosate, does not pose a threat for humans. And there have been lots and lots and lots of studies over decades looking at the effects of glyphosate on test animals and on cell cultures. Oh, and then about the lawsuit. Yeah, well, the problem is that, you know, a jury is not made up of scientists. A jury is made up of peers. And I would guess that probably during jury selection, most scientists are taken off by, during jury selection, and that you're left with people that don't have a good understanding of biochemistry necessarily, and that they are going to be swayed by whichever lawyer is most persuasive. And that's the side they're going to go for. So it doesn't have anything to do with science. It has to do with human nature.
Yes, playing into human emotions.
That's right.
Okay. Well, that was an amazing presentation. We learned so much, a lot of food for thought. Definitely going to go be more careful with what I read and tell people. Any last questions? I will send out the recording and the handout, Linda's contact information. Again, she has a bunch of links and websites for you to check out should you wish. Thank you for your time, everyone. I guess I will let you guys go and have a good Friday. And Linda, thank you so much for your time.
Oh, you're welcome. Thank you, Sophia, and everybody else. I enjoyed starting my day off with the ability to get on my soapbox and rant about stuff.
Well, we enjoyed it.
Oh, good.
Thank you so much.
You bet. Bye-bye.
Right, bye. Bye, everyone.